Our next speaker, I believe, is joining us online and will be projected shortly. It's Professor Kelly Bolton, who I have not had the pleasure to meet, but I know of her by reputation and reading her papers. She's assistant professor in the Division of Oncology at Washington University. She's a physician scientist at Washington University, and she works within the stem cell biology division. Her research focus includes cancer genetics, epidemiology, genetic susceptibility to cancer, and she wrote a really important paper in 2012 about the survival of women with BRCA1 and BRCA2 mutations with ovarian cancer. It was a really important paper then, and she's also published widely on clonal hematopoiesis. But she was the first author on a seminal publication in clinical cancer research in late last year, November 2022, in Clinical Cancer Research, entitled Molecular Subclasses of Clear Cell Cancer and Impact on Biological Behavior. And she's going to be talking about that today, and I really look forward to hearing her speak. Thank you very much, Kelly.
It's 2 a.m. where I'm calling from, so I hope that I do a good job presenting. But I was just so excited to get this invitation to speak with you guys. This is a really amazing lineup, and I'm excited to share our work. We started off, just deciding that we wanted to profile clear cell ovarian cancer quite a few years ago, and the reason was because clear cell ovarian cancer is a pretty common form of ovarian cancer, second most common. As you guys know, it has a really unique spectrum of mutations compared to high-grade serous ovarian cancer, but pretty poor outcomes when it's diagnosed in an advanced stage. And there's been really fantastic work from the people in this audience and the presenters looking at how molecular subtypes of high-grade serous ovarian cancer predict response and echo biologic differences in the important pathways in tumor initiation and growth. But unlike high-grade serous ovarian cancer, this really hasn't been done in a large way in clear cell ovarian cancer. And so our question was, really, could we use molecular subclassifications of clear cell ovarian cancer to help us inform personalized medicine, and do these even exist? And so to achieve this, we performed a multi-center study, including samples from Anna DeFazio at Westmead Hospital, and it took us many years to get this group together. In the end, it included 10 sites, almost 450 tumors. We did a mixture of central DNA and RNA extraction and local extraction. And when possible, we did a central path review of slides at MSK. In the end, we had 420 samples that passed QC. So, the largest cohort of clear cell ovarian cancers that had been genomically profiled as of this time. And we had clinical data and survival data on most, but not all of these cases. So we performed targeted DNA sequencing on these. And the reason for that was really just cost. There were some pretty big studies that had looked at whole exome or whole genome profiling of clear cell ovarian cancer, including some public databases for this. And so we had a sense of what genes probably were going to be the most important. So we targeted 163 genes that had been recurrently mutated in clear cell ovarian cancer based on this previous published data or public databases. We designed this to include widely spaced SNPs to capture copy number events. And we sequenced this very deep, down to about 500x coverage. The reason being because we wanted to be able to detect both, clonally dominant, but also highly subclonal mutations in these tumors, as we know that both of these factors are important in determining outcome in other cancers. And so we did variant calling using a consensus approach with multiple variant callers. And because of our depth, we were able to detect mutations reliably down to about 2% VAF. And in parallel on these same tumors, we had about 200 where we had really high quality RNA. And we profiled these using whole transcriptome sequencing using poly-A capture. So using this targeted panel, we found at least one driver mutation in almost all of the tumors, so about 95%. As has been described in other cancers, the median number of mutations was around 3, with a range from 1 actually to 25 driver mutations. As you can see on the left, the most frequently mutated gene was ARID1A, followed by PIK3CA and mutations in the TERT promoter. And on the right, I'm showing the frequency of these mutations plotted against the variant allele frequency. So, mutations that are at higher variant allele frequencies presumably are more clonally dominant or earlier events compared to mutations that are present primarily at lower VAFs that are likely secondary events. So we saw that, mutations like ARID1A and TP53 were much more likely to be at higher VAFs or early events compared to other genes like CRE, BP, which were probably more likely to be subclonal. One interesting thing that you might be picking up now is just the relatively high proportion of tumors with TP53 mutations. We reported this at around 16% of our cases. This is in line with some reports. I'd say the median number of TP53 mutations from prior studies range between around 9 to 18. But on average, it was a bit higher than other people had reported. So we looked at this a bit more because we were wondering, what could be causing this higher proportion of TP53 mutations. And one, I think, is differences in sequencing depth. We sequenced very deep compared to previous studies that were using a more whole exome, more whole genome approach. And about 14% of the TP53 mutations we detected were under 10% VAF. So these really wouldn't have been previously detected using lower depth sequencing. But the second possibility we wanted to really explore was whether these could be misclassified high-grade serous ovarian cancers. So to look at this further, we took a subset of the cases where we had FFPE slides available, and Martin Coble actually stained these for IHC and then provided a morphology review as well. And indeed, he saw that about a fourth of these cases were misclassified. Out of the 14, 3 were endometrioid carcinomas, and there was 1 high-grade serous ovarian cancer. So extending this further to our whole cohort, if we extrapolate that this amount of misclassification applied to the rest of the tumors, we would estimate that around 19 of our 71 TP53 mutant tumors were probably misclassified. So we think that misclassification is involved, but probably couldn't explain the vast majority of our TP53 mutated tumors. So I'm going to now move on, and why I'm harking on this will make sense more later as well. So we looked at how our most commonly mutated genes by SNB and Indel might relate to clinical outcomes. since we know for high-grade ovarian cancer and other cancers that there is differences in presentation by tumor genetic background. And so we looked first at history of endometriosis and Asian ancestry, and we saw that individuals with ARID1A mutations were more likely to have a history of endometriosis, with a trend towards individuals with TP53 mutations less likely to have a history of endometriosis. And similarly, we saw that individuals with an ARID1A mutation were more likely to be of Asian ancestry, although this wasn't significant after multiple testing correction. We also saw that individuals with TP53 mutant tumors were more likely to have advanced stage disease, whereas other mutations were, more equally distributed across advanced stage and early stage disease. Next, because we noticed that, most of these mutations are not occurring alone, but most people have an average of 3 mutations. We wanted to understand whether there were patterns of mutual cooperativity or exclusivity between these driver events. So we looked at the pairwise association between different mutations, and that's shown in this heat map. So the frequency is shown in the bottom, with more frequent pairs being darker purple. And then the odds ratio for the pair occurring together is shown on the top. If things are likely to co-occur together, that's shown, with an orange. Things that are mutually exclusive are shown in blue, and things that are significant after multiple hypothesis testing correction are shown with an asterisk here. So we saw statistically significant co-occurrence between ARID1A, PIK3CA, as you can see over here, but we also saw mutual co-occurrence between TP53 and BRCA1 and BRCA2. We saw mutual exclusivity between ARID1A, TP53, and PIK3CA and PIK3R1, suggesting that these probably represent distinct pathways to cancer initiation. And because, clearly there are some mutual cooperative and exclusive patterns that we're observing here, we performed unsupervised clustering analysis to try to define non-overlapping subgroups of clear cell ovarian cancer based on an even broader, beyond just pairwise, mutational co-occurring patterns. So using this approach, we identified 7 subgroups of clear cell ovarian cancer, and we compared the frequency of mutations between subgroups. So here I'm showing the various clusters, and then the frequency of mutations among these clusters. So the most frequent cluster was cluster F, and this was defined by mutations, for example, in PIK3CA and TERT. So you can see that I'm going to go through by now, instead of descending order, frequency of ARID1A mutations. So cluster A was characterized mainly by an ARID1A mutation in combination with other common clear cell ovarian cancer mutations like PIK3CA, TERT, or TP53. Cluster B was characterized by an ARID1A mutation in combination with a less common disease driver mutation, like P10 or ATM. Cluster C was characterized by, again, an ARID1A mutation with PIK3CA, Cluster D was characterized by multiple ARID1A mutations and PIK3CA wild type tumors. And then we saw two clusters that were ARID1A wild type. Cluster E that was dominated by a TP53 mutation, and cluster F that was defined by other non-TP53 disease-defining mutations. And finally, we saw this interesting cluster of individuals that were characterized by mutations in SMARCA4, a mutation, as you know, that's typically observed in small cell ovarian carcinoma. We also, I should mention, performed pathologic review, or Martin Coble performed pathologic review of these cases, and he decided none of them were misclassified. One interesting difference maybe between this cluster of tumors and small cell ovarian carcinomas are that this cluster, is dominated by the SMARCA4 mutation, but also had other co-occurring mutations like PIK3CA, TERT, TP53, unlike small cell ovarian cancer, which tends to not have that same co-occurrence of mutations. So now that we have these broader tumor subtypes, we wanted to again explore how these might vary based on clinical presentation. So we looked at the same things we had looked at previously: age at diagnosis, tumor stage, history of endometriosis, and Asian ancestry. So in terms of age of diagnosis, the only clear pattern we observed was that that SMARCA4 cluster seemed like it had a younger age at presentation. On average, people were in their mid-50s. Again, we saw that the TP53 mutant tumors were more likely to be advanced stage at presentation, but we clarified this to explain that it was not so much just TP53 mutations, but it was the TP53 mutation in addition to being ARID1A wild type that really defined a group that was more likely to be advanced stage. Similarly, we saw individuals with ARID1A mutant tumors across the molecular subgroups were more likely to have a history of endometriosis and were more likely to be of Asian ancestry. So next, we wanted to understand how transcriptomic profile might correspond to these molecular subtypes, and how we might use transcriptomic profiling to maybe even just describe independent subgroups of ovarian cancer, of clear cell ovarian cancer. So we performed unsupervised clustering of our RNA-seq data and identified two main RNA clusters, which I'm referring throughout the talk as cluster 1, which is the dominant cluster, and cluster 2. So we saw that when comparing RNA, the whole transcriptome RNA cluster 1 to cluster 2, that expression of that cluster 2 was enriched with tumors belonging to mutation cluster E. That's TP53 mutant, ARID1A wild type tumors, with the rest mainly coming from this undefined mutation cluster. We looked at the most highly or differentially expressed genes between cluster 1 and cluster 2. And we showed, and that's shown over on the left. So cluster 1 showed higher expression of genes that have been previously reported in clear cell ovarian cancer, including ANX-A4 and GPX3, both of which actually are linked to platinum resistance. Among the most highly expressed genes in cluster 1 compared to cluster 2 or also genes that are known to be overexpressed in endometriosis. And when we performed pathway analysis of the overexpressed genes, some of the most important pathways that popped up were flavonoid, glucuronation, and monocarboxylic acid metabolism. Boy, that's a lot for 2 in the morning. Expression cluster 2 was enriched for expression of genes involved in ECM organization and mesenchymal differentiation, and also some genes like WT1, which are features that are again classically associated with high-grade serous ovarian cancer. So finally, we wanted to look at how our molecular, how individual mutations and molecular subtypes of clear cell ovarian cancer might relate to disease outcomes. So we looked first at how individual mutations might associate with overall survival, and we found a trend towards in univariate analysis that ARID1A mutant tumors might have improved survival compared to TP53 mutated tumors, although this was non-significant. But because within our mutational clusters, we saw really similar behavior of the ARID1A mutant clusters, but very unique presentation of the TP53 mutant ARID1A wild type tumors. We used those 2 broad clusters to look at differences in survival. And so we saw that in univariate models, that ARID1A wild type TP53 mutant tumors shown in green had worse overall survival compared to ARID1A mutant tumors. Similarly, RNA cluster 1 showed a worse showed an improved survival compared to RNA cluster 2, which again represents the TP53 mutant ARID1A wild type tumors. So, however, once we adjusted for stage, this was no longer significant, and the effect really diminished. So a lot of this worse survival appears to be driven actually by tumor stage, with individuals of similar stage having similar survival. And we even saw a trend towards the ARID1A mutant tumors actually having a worse survival after adjusting for stage. So the final thing that I'll describe today is this kind of a brief analysis where we looked at molecular subtype in response to platinum. So we wanted to understand if some of these divergences in our survival analysis after adjusting for stage might be accounted for by differences in the response to platinum. We did not have great data on response to platinum in the cohort. It was only available for a subset. But so among women who had advanced stage disease, who had successful debulking and a known response to platinum, we looked at how these molecular subtypes might relate. There were only 36 people, so this is very exploratory. But in general, we observed a higher proportion of ARID1A wild type TP53 mutant tumors that achieved a CR. So it was 1A mutant tumors that was only around 55%. So interesting observation, and might in a larger cohort be significant. So in summary, our results of this integrative genomic and transcriptomic analysis suggests that there's 2 main subtypes of clear cell ovarian cancer. The first, which represents about 83% of clear cell ovarian cancer, we called the classic CCOC, where you observe a mutational profile that includes ARID1A and other common CCOC drivers like PIK3CA and TERT. We saw enrichment of expression of metabolic pathways. These cases tended to present with earlier stage disease and were more likely to have a history of endometriosis. And the second subgroup, which we call high-grade serous ovarian cancer, represented 17% of our cases. This was characterized by a TP53 mutant, ARID1A wild type tumors that showed enriched expression of genes involved in ECM organization, immune-related pathways. These tumors tended to present at advanced stage, be of white ethnicity, and we saw some evidence that they might be more likely to achieve a complete response following first stage platinum therapy. So what we think might be interesting things to look at moving forward is this idea that the classic CCOC group, if they are relatively platinum resistant, might be the group that one would want to target among clear cell ovarian cancers for investigational frontline therapies. There's several kind of promising agents that are being explored now, like immune checkpoint inhibitors, PI3 kinase inhibitors for PIK3CA mutated tumors. Future work, I think clearly it would be important to increase our genetic diversity, particularly of women of Asian descent, East Asian descent, who we know have higher rates of CCOC in general. And last, we hope that this study can provide justification for additional work like this, defining molecular subtypes of ovarian cancer, and hopefully can be used by you and others to justify this work and to show that it can lead to clinically meaningful molecular subtype identification. So thank all the collaborators on this study. There were so many, I got lazy and I just included all the authors on this slide. I want in particular to thank Martin Coble, Ellen Good, Kate Lawrenson, whose group performed some of the RNA-seq analysis, Jason Connor, who provided funding for the study, and Ellie Papa-Manuel, whose group I worked in when I was doing the majority of this analysis, and has supported the analytic aspects of the study. So thank you, and I look forward to the Q&A.
So we've had a great session today. Unfortunately, we only have about 5 minutes. Maybe the organizer will give us a couple minutes more, but because of all the sessions, we can't go on for too long. So it's and ask people to stand up and ask any questions. Are there any? Okay, question from Claire to Kelly.
Comment on those very low frequency, so low mutation cases. If it was under 10%, I'm sorry if I missed it, where did they fall out in your analysis? What's the indication of that very low mutation? Hear me? Do you want me to repeat it?
Yes, I'm sorry. I couldn't.
I've just got the microphone. So in your where you've discovered these low allele frequency p53 mutations, less than 10%, so that might help us understand some of the cases better. But what was the clinical outcome of those cases where it's such a low allele frequency? Do you think it's relevant?
Yeah, I don't think I didn't look, I have to admit, I don't think that I did an analysis excluding the TP53 mutations. Maybe like one observation that I'll share, which might be relevant, is when Martin performed the histologic review, interestingly, for some of the tumors that we sent that were TP53 mutated, he didn't observe that they actually had IHC loss of TP53. So presumably it was not biallelic TP53 loss like you see in high-grade serous ovarian cancer. Probably these cases were much more likely to just have a single TP53 mutation, which in other settings has been shown to alter disease outcome compared to biallelic TP53 mutations. So there probably is, like you're alluding to, Claire. I mean, you know, if we stratified these cases as to whether they were biallelic TP53, clonally dominant TP53, we might be able to get it even further, you know, molecular like subclassification of those tumors, which would be really helpful.
Great, thank you very much.
David?
Hi, Kelly. David Botel. Terrific study.
Oh, thank you.
Two questions quickly. We don't feel that BRCA mutations occur very frequently or even perhaps at all in B-cell cancers, but there have been different views about that. Did you see any BRCA mutations? And the second question is a sort of more general one. Did you expect things to be more transcriptionally different between cancers with different drivers? It sort of almost seems like all roads lead to the same transcriptional phenotype in a way.
Yeah, I agree. I know. It was a little honestly disappointing when I did it. I thought we were gonna find all these like transcriptomic you know, subgroups, and it was all just like TP53 yes or no. Maybe, I mean, you know, it was a small cohort. I don't know maybe how much you would expect to see additional subclasses with 200 tumors. But the most dominant, you know, difference was between the TP53 and mutated and wild type tumors. We did, you know, unfortunately, we did not perform matched blood and tumor sequencing for a lot of these cases just because of cost. But we did observe BRCA1 and 2 mutations. Some of them were like founder mutations, so they probably were germline. Most of them were not. So kind of hard to tell if they were germline or you know, just somatic events. That would be, I would say, you know, in the next study, if this were to, you know, be expanded, that would be really important, probably, to get more at the germline background and how that was influencing, you know, the tumors presentation and outcomes as well, like you're alerting to.
Do you recall the frequency of the ones that you thought were founders?
Oh, it was like
Not at 2 a.m.
just a couple. Honestly, it was like a couple of tumors that were like where it was where we were looking at it. And I was like, oh, this is probably a germline mutation, but most of them it was hard to tell.
Thank you.
Thank you,
John. I think we've got time for one more question. There's John Zaltzberg in the back.
Thank you very much for a great session. Just a quick comment. We've got 140 clear cell tumors, patients rather, in the database with clear cell tumors, if that would be of help. The question, though, I was going to ask David and Claire, are we any closer to having a drug that might target TP53?
No, but there's a lot of work going on. I mean, I think just as a final comment, I mean, it's a really important question, and there is obviously enormous activity in that area, but a final comment before we head off for tea is we almost have to go back and do all our trials again, because we love simplicity in medicine. And everyone gets lumped together. We do all the science, we do the biology, we understand they're all different and they're multiple subsets, and yet we then design trials where we call it high-grade serous cancer, clear cell cancer, whatever. And so all the studies that were done, for example, with clear cell, looking at mTOR inhibitors, and then we said they had no activity in clear cell cancer. We have to go back again and actually subdivide and categorize the subset that may possibly benefit. And the same goes for surgery, because we lump everyone together. We know they're all different. And so my concern with some of the studies you just alluded to, where we're going to give 8 cycles of platinum-based chemotherapy as neoadjuvant, potentially, to people who've got BRCA mutations or HRD, and I can think of no better way of inducing resistance and BRCA reversion mutations than giving a whole lot of platinum. and then giving them a PARP inhibitor afterwards, and then asking ourselves, why isn't it working so well? But we're lumping them all together. And I think this is just something for us to ponder about, because this could be a meeting in its own right. How do we design trials moving forward and moving away from the one size fits all when we know it makes absolutely no sense? And with that, enjoy your tea.